So the next speaker is Alessandro Gozzi. He has a master's in molecular biology and a PhD in biomedical imaging. He's a senior scientist and principal investigator at the Italian Institute of Technology in Italy, where he has the functional neuroimaging laboratory. Research in the Gozzi lab is aimed at understanding how the brain works as an integrated network, both in healthy and in pathological states.
Okay, thank you so much for having me here. It's a great pleasure to present my research to this audience. This is going to be a little bit weird, I guess, because I'm going to show you a lot of pictures of mouse brains. But I guess this, well, hopefully, will make sense in the end. And so basically, this is just to introduce the goal of my talk, which is basically to try and understand mechanisms, to decode and somehow infer, to the best of our capabilities, what differences in connectivity, here they are simply conceptualized as hyper or hypo connectivity, can be translated into testable hypotheses, right? Because that's one of the big goals of the field. So, and I'm going to start to contextualize my research by giving you a very quick refresher. Sorry, I'm just trying to understand how this goes on. Green? Okay. So I'm going to give you a quick refresher of very basic concepts you all know, but they are very important to understand what I'm going to show you, okay? And so clearly, I'm not going to explain what functional connectivity is to this audience, you're experts, but the key point I want to emphasize with this specific slide is that functional connectivity is a measure of synchronicity. Keep that in mind. That's very important to understand what I'm going to show you onwards, okay? So synchronization between this spontaneous fMRI signal across regions. The second important point I want to refresh, and this is like a given here, of course, it's what's reported in this slide here. So fMRI connectivity is altered in pretty much every brain disorder. However, I think we all agree that the underpinnings, right, the physiological basis and the clinical significance of these changes are unclear. We don't really know what they mean, right? We also seen it in the previous presentations. And so I think the key postulate of my talk is that our inability to address these important questions mainly stems from the fact that there's this like big explanatory gap between models of brain function at different levels of inquiry, right? We can study, cellular mechanisms or connectivity at the microscale. We can study very well, of course, investigate systems level, but we have trouble when it comes to bridging these fields. And so what we've been doing in the lab, actually, our proposition is that one way, not the only one, but one way to narrow this gap is by perhaps going this way. So basically, you can implement system measurement of whole brain activity in physiological accessible species, such as the mouse. That's what we've been doing. And by doing that, we got two major advantages. First, we can begin to understand what the mechanisms are. And second, we can also somehow establish causality between events. And that's a very important thing, because in human research, there's a lot of correlation, right? It is difficult to establish causality. That's what we've been doing in my lab for in the past 10 to 15 years, so we pioneered, fMRI mapping in mouse models. And so my research today is very strongly influenced by these very busy slides. And I'm going to explain it, bit by bit. And I'm not going to tell you the whole story here. If you want to understand what this is about, go and check out the talk of my student, Sylvia, who's here in the audience. But, this slide is basically a summary of what we've been doing as part of a big project. This is still unpublished, when we try to map connectivity changes introduced by autism-associated mutations in 20 different mouse models. So these are 20 different mutations. And why am I showing you this at all? Well, there are two interesting things, in my opinion, that I have to deliver. First of all, this is just a technical communication, if you like. So from now on in my talk, when you see red, okay, these are connectivity differences. When you see red, that means hyperconnectivity. Keep that in mind. And when you see blue, means reduced connectivity, hypoconnectivity throughout, okay? So this is just to introduce my color coding. The second important thing here, if you look at these connectivity differences, there's a bit of a spectrum, right? A little bit of a mix, red and blue. However, you know, you don't need fancy machine learning to realize that there's a bit of a polarization here. These differences tend to polarize into two dominant signatures. It doesn't matter what they happen here in the brain. It doesn't really matter at all. But as you can see, these can be grouped into hyper and hypo, okay? And this is important because this is something that prompted our research to say, you know, these are like two diverging sort of changes that we see, you know, prominently mapped in this landscape. And so the question is, what does hyperconnectivity and hypo mean in terms of physiology, okay? And so we try to address this question. So the problem here is, where do we begin, right? It can mean many things. Well, I think a good start to place is here, right? You all know this, right? The structure constraints function, right? So functional and structural connectivity are closely related. This has been shown in humans, so I'm going to show you my own research just to change tune a little bit. And you know, this is just a modules of the axonal connectome of the mouse, and if you compare them spatially with fMRI modules, you see they are the same, right? This is something you all know. And so, as I said, this is a good place to start. Why? Well, clearly, if a structure drives function, right? In this situation here, I told you blue is less connectivity. Perhaps we have like some sort of a lower input, lower axonal input, okay? And this is something perhaps obvious, like trivial. Everyone could think of that. But interestingly, a causal demonstration of this relationship are lacking. And so in the lab, we say, okay, why don't we just start here? We can use the mouse, and we can be causal in this case. We can somehow silence, reduce activity in one region under the assumption that by doing that, we'll see less connectivity, right? And that's what we did a couple of years ago. And clearly, I'm not going to take you into the methods. This can be done in rodents. You have to trust me and also the reviewers, because this paper is published now. But basically, here, what we did, we used two different ways of silencing, so reducing activity in it's a key hub of the mouse brain of the default mode network. By the way, mice do have default mode networks. And basically, we silence the prefrontal cortex, medial prefrontal cortex, chronically or acutely. And the perk, our interest here in, and perhaps the advantage of this approach is that in mice we can also study electrophysiology and so relate fMRI to electrophysiology, which is pretty interesting, I guess. And I hope you find it interesting too. So what have we found? Okay, so first of all, this is chronic silencing, okay? We overexpress the sodium channel to reduce excitability, so we are silencing output. And okay, so how do we interpret these maps? It's easy. So this is like a seed-based map of control mice here on top. And this is what happens when you chronically silence, which is kind of weird, right? Because these maps, they look very similar. And in fact, there's more stuff here. And when you map a difference, much to our surprise back then, I still remember to this day, we found that there is more connectivity. And if you're wondering, these regions that show more connectivity are regions that have strong projections from the prefrontal cortex in terms of axons. So this is interesting, but as I said, also a little disconcerting. And back then, I told my student, get out of here. This is impossible. Yeah, of course. This must be some sort of, like a compensatory effect. And so we said, we have to resort to something that can give you like a real-time silencing. Let's go with something that is really real, right? So we said, okay, let's replicate this with something that is a little fancier. This is called chemogenetics. It's not very important what it is. It is like optogenetics, which I assume many of you heard. But instead of using the light to silence or stimulate neurons, you can use these synthetic receptors, okay? You express them in the mouse, and then you can give a drug, which is an inert drug, that binds to this receptor, and by doing that, you have this like, real-time effect, either silencing or manipulating your neurons. And this time, we didn't want to take any chances. We say, okay, let's take a look at what happens, and let's start with electrophysiology. Are we really silencing the region? Yes, we are. Okay, you can see this is just the experimental group, and neuronal activity is going down, so cool. Of course, I'm going to also show you here like a spectral analysis with electrophysiology. You can see here that blue means reduction in high-frequency rhythms. So all well. What happens to connectivity? Well, here we can really see it, real time. Now, where is the pointer? I lost it. But anyway, it's up here, right? And it goes up. So unequivocally going up. So it's not a compensatory thing. You silence the region, you have more connectivity. And if some of you are wondering, oh, maybe, preprocessing, this sort of stuff, we've seen papers with 1 million of preprocessing pipelines. We also try here, where is it? Yeah, with global signal regression, the fact is still there. It's not a global effect. And okay, so that's perhaps interesting, right? Because, well, at least to me, because part of the title now may begin to make sense to you. So less, perhaps, is more, right? Less activity, so silencing is more connectivity. However, it is, as I said, a little disconcerting because, what is going on here, right? There are many questions. And there are two questions I want to carry over now for the rest of my talk. The first one is this a generalizable phenomenon? Because clearly, many of you are wondering now, right? Or specific to this specific manipulation on area. And the second important thing is, if it is, spoiler, it is, I guess, it's what the heck is going on? Why do we have this? So let me go back to the first question. So is this generalizable? Well, if this is generalizable, then when I do the opposite of silencing, which is increasing activity, I should have the opposite of hyperconnectivity, which is hypoconnectivity. That's exactly the hypothesis we tested here. So what we're going to do now, what I'm going to do now is going to show you the results. So this is unpublished, where basically we carry on this research in this direction. So we increase the excitability of the prefrontal cortex, and this can be done in two ways. One, you can increase excitation, so pyramidal neurons, they fire more. And the second way is by reducing inhibition, right? And as I said, I didn't want to take any chances. Both of them we did. Okay, so what have we found? We increase excitation here. Electrophysiology first, because we don't want to, to take any risk. We want to make sure everything is working. So you are familiar with these curves now, right? This is firing rate over time. This is a quantification. It's going up as predicted. And this is the corresponding spectral signature. So you have a lot. Where is it? Where is it going? Okay, it's here. So we have a lot like, high frequency activity, and you have, of course, these neurons are firing non-synchronously. You also have like lower oscillatory activity because neurons can either, fire in synchrony or not. But so far, so good. This is a little technical. What happens to connectivity? Well, as I like to say, well, you can see for yourself, right? This is control, this is excitation, and as I like to say, this is the very first time ever in my career when my hypothesis is correct. As a matter of fact here, not really, the very first time. So connectivity is going down, okay? And some of you are perhaps wondering, we might sometimes use sedation, but we also can do it now awake, and also in awake conditions, we have blue, hypoconnectivity, right? Okay, but as I told you, we are interested in generalizability here. So if it's generalizable, it should also apply to reduce inhibition, which is another way of increasing excitability. So let me go on. Again, you're familiar with this graph. Now we are reducing inhibition. You can see that shrinking. So firing rate up, all well. And what is interesting here, look at the spectral signature. Here you have a lot more oscillatory activity, which kind of makes sense if you're into physiology a little bit. Still, you have more firing rate, which is what we wanted. What happens to fMRI connectivity? Can it be the same in spite of this different spectral signature? Well, as a matter of fact, it is. We still have a reduction in connectivity, okay? And just to take a little sum up before I wrap this up and explain perhaps what happens here, or at least how we can interpret this. So the sum up is so far is, as I said, there's an interesting point, which is the title of my talk. Kind of makes sense because I show you more activity, right, like here, less connectivity, and the other way around. But this is also interesting, and as I said, and maybe bizarre, because how can we now reconcile this different spectral signature with what, what the fact that connectivity goes up and down, right? What is the model underlying this? Okay, so in the next 2 minutes, I'm going to explain, hopefully, what we found and what we come up with. How can we try to? This is very important. I'm going to ask you a last effort. So what we try to do here is to understand this in terms of electrophysiological connectivity. So here we put one electrode in the region that we silence, which is this one here, the prefrontal cortex, and a region that is strongly innervated, but it is the posterior cingulate, basically. It's called retrosplenial in mice. And this graph here is a key graph. This one is electrophysiological connectivity. It's technically is LFP coherency. Across frequency, you can see, because with electrophysiology, you can span all the frequency. And what I'm showing you here is the difference. So it is the effect of the manipulation. So basically, this could be a little busy, but it's very simple for you to see that when you do electrophysiological connectivity, and this is just reducing inhibition and excitation, these two connectivity profiles are very different, right? And yet, we got this, with these two manipulations, yet we got the same fMRI connectivity signature. So how? How can we reconcile this? Well, one way of reconciling this is by assuming that fMRI is very sensitive, and in fact, it is disproportionately sensitive to the very slow part of this spectrum. I'm going to help you see it here, okay? And I'm going to help you even better here. Look at that. This is just the same graph, log scale. If you focus on this, and fMRI is indeed an infraslow mechanism, right, an infraslow fluctuation, here the difference, so this is just the difference between control and manipulation, is negative. So we have lower electrophysiological connectivity. And so this is clearly a hypothesis at this stage. But if this hypothesis holds, then you remember my previous manipulation when I silence the region. When I do the same analysis on those data, I should have stronger coupling in this frequency area. And this is exactly what I get. Look at that. Here, it is strongly increased. And if this one is driving fMRI, I expect, the differences in fMRI coupling that I just showed you to covary with difference in LFP, so in electrophysiological coupling in this band, but not in other bands. And that's exactly what we found. This is my last slide, basically. We found that basically the amplitude of fMRI is predicted by changes in slow and infraslow coupling, but not in the other bands, okay? And so just to sum up, because my time is over, this is the model that I throw at you, and I'm very happy to hear criticism and or suggestions or praise or whatever. And there are two things that I want to deliver. And I think I said it over and over, that remember that based on our results at least, fMRI connectivity is disproportionately sensitive to slow frequency, electrophysiological frequency, and much less sensitive to faster rhythms. And the second thing is this, the rule of my title, basically regional firing activity, which is, also a measure of excitability, is inversely related to fMRI connectivity. So less is more and more is less. I think I'm going to stop here. I have clearly to thank all the people in my lab, all the funding agencies. And, well, we are recruiting. Probably you're not the right audience. But, yeah, thanks for listening.
Okay, we have time for one question.
Hi, that was a fantastic talk. When you are increasing the excitability or decreasing the excitability, in a sense, and then looking at resting state fMRI, the neurons aren't necessarily responding to some kind of stimulation or some kind of cognition. And so the increased or decreased firing might just be related to some sort of like increase in noise. Do you think that might potentially explain why there's some of these effects? That's exactly what we mean. Yeah, that's and this is really referring to autism or developmental disorders where there's these baseline changes, especially in inhibitory activities. So that's one way to go forward. Here, this clearly does not apply to entrainment, right? If you entrain a rhythm, you will see more connectivity. But this is just baseline, and that's resting state, which many of you are using. So at least we have testable hypotheses. Yes, of course.
Okay, in the interest of time, I will invite the last speaker to the podium.